you. So hopefully no one misunderstood that I'm not going to talk about physics. I almost failed out of high school for physics, so it's not about like harmonics or whatever. It's about also not like this kind of waves. I think we all want to be there, but maybe we'll just look at that picture. What I want to talk about is the wavy structure that exists in many tissues in our body. So this is fat, lung, ovary, lymph node, some zooming pictures, ligaments, and arteries. Most of these pictures show collagen fibers. You can see that some of them are aligned, like in arteries and in ligaments. Others are more randomly oriented, but you can all appreciate these wavy structure that exists in these fibers. I will talk a little bit about why, or why we think they exist. And these wavy structures are often lost in disease and in aging. So in the picture to the right, next to a mammary fat, is breast tumor, breast cancer. So the dark area is where the tumor is, and you can see the white lines are the active the collagen fibers that have been straightened. Most likely due to the cancer cells and the surrounding cells pulling on these fibers and straightening them, aligning them. And I will talk about the artery part in a little bit. So why do we have these wavy structures in our body? In mostly the connective tissues, that's somewhat mechanically oriented, like it has a lot of mechanical function. So the two tissues that my lab focus on are ligaments and tendons, which are very similar, and arteries, or in this case, aorta. Again, the wavy structure in ligament is collagen, and the wavy structure in aorta is made mostly of collagen and elastin fibers. And you can appreciate in these, the red dots here are cell nucleus. Here is also nucleus, and the green here is cell skeleton. So the cells that live in these tissues also follow these wavy structures. So the function of the existence of these structures is mechanical. You can imagine they basically are like springs. So a spring can be made of, say, stainless steel. But because of these wavy structure, you can maybe even use a finger to push on that spring and it would deform. But if you stretch the spring long enough, you're engaging in the metal itself, then it becomes very stiff. So that's how it works in our tissue. When the waves still exist, you can pull or push on the structure at low force and it would deform. So you can imagine in our ligaments, I can move my joints pretty easily at this low strain level. But at a higher strain level, if I want to straighten my arm, that ligament will be stretched to this sort of linear region. So you need a higher force to straighten that tissue. So in terms of ligaments and tendons, your joints are locked, so you don't dislocate your shoulder, for example. So that's a crucial, this sort of nonlinear mechanical function is a crucial component of, or crucial reason why these waves need to exist in these tissues. And so what happens when they're damaged? So these are cross sections of arteries. So you can see the sort of dark or black lines in both pictures are the elastin fibers, which exist in the medial there of our arteries, if you know your anatomy, but that doesn't matter at this point. So you can see the waves in a healthy artery, and in the injury artery, you can see the dark lines are straightened and even fragmented. So the elastin fibers are damaged. Moreover, you can see this very thick pink layer in the lumen of the artery. So that's your cells trying to compensate for the loss of mechanical function and synthesize a lot of additional matrix, which is usually collagen. So who makes these switches? It's the vascular smooth muscle cells that live in these wavy structures. So again, in a healthy wavy structure, the cells are quiescent but have high contractility. So they're muscle cells, right? So muscle cell. So they contract and control your blood flow or the tone of your blood vessels. But when they undergo this so-called phenotypic switch, they lose that quiescent and contractile phenotype and become synthetic. They will proliferate a lot. They would make a lot of matrix, synthesize a lot of matrix, and change the structure of that matrix. So this is obviously extremely simplified, but they have this correlation of the changes in the VSMC phenotype with the structure. So our question in both arteries and ligaments and tendons is, how does this mechanical environment, mechanical structure, regulate these cells? Is it merely correlative as we observe in histological sections, or is it a causal relationship? So to do that, we need to be able to recreate that environment, because you can only find correlation in humans and in animals. So we use a technique called electrospinning and use polymers basically to generate these fibers. We can make them straight and aligned, and with some processing, we can make them wavy. A professor in chemical engineering saying it's basically like perming your hair. You can make your hair wavy, you can make your fibers wavy. So with these wavy structures, you can see in the EM pictures, we can find that there's a corresponding changes in mechanical property. So I think that's blue. The blue line is represent the stress-strain relationship of the straight fibers. So you don't see much of that, so-called tow region, the low strain or the non-linear mechanics. But with the wavy fiber, we can see there's this, it's responding very slowly, this sort of tow region. So you can pull on this, you can deform this fiber or material relatively easily at low strain, and it becomes stiffened with high strain. So we can recapitulate the mechanics of this tissue, but what do the cells do? So the two things the cells could see in terms of physical environment. The first is its structure, the other possibility is mechanics, right? So let's first look at the structure. So if you just put cells on there, oops, I only have 3 minutes. The cells can follow the structure, the cells look different, and they have different expression. So type 1 collagen expression, for example, is increased. And if we subject these cells on this fiber to a dynamic stretch, we can further change their gene expression. So on a straight versus wavy or crimped fiber, and in the red groups, they are subjected to exercise, basically, simulating daily exercise. We can see more than 30 times more collagen gene expression. So this is a dramatic change, meaning these cells can sense both the structure and the mechanics of this tissue. So how do cells even know shape? How do cells know that they've been stretched? So one part of it is to understand the so-called strain transfer process. The cells in the tissue or in the material is sensing a different mechanic. So these dots are cells and they're in different fibers. So you can appreciate the difference in the strain field, in the formation field in them. And you can also see the cell. If you label the cell body, you can see the cells, instead of being elongated, it's being sort of straightened. So we believe these are different signals to a cell. And that's probably how it happens when we exercise our cells or when our heart pumps. So this is done in collaboration with our friends in Penn, and they did some mechanical modeling. But to understand what happens in the cell, right? That's outside the cell, but what happens in the cell? Those fibers you can see are somewhat heterogeneous. So to have very precise control of cells, we would use microcontact printing or microfabrication techniques to control cell shape. If you want, we can write your name. Lots of people do that 20 years ago. With this precise control of cell shape, we can have more measurements in cells, single cell measurements. So we can measure intracellular forces with this traction force microscopy. We can stretch them on a microscope in a precise shape and look at the intracellular strain field and even maybe intranuclear strain field. And we can also look at the cell's life. For example, these cells are moving. We can appreciate the cell skeleton shape changes and nuclear shape changes, and hopefully one day chromosome changes. So I think this is an important slide. What we're interested in is cell morphology, cell structure, and even anisotropy. I didn't talk about it today, but these things are aligned. So we all know anisotropy. So how does the cells know direction? How does it tell from X and Y and Z? How does it interact with the physical stimulation, dynamic physical stimulation, basically exercise? Our model systems or tissues are ligaments and arteries. But if you study other systems with this wavy structure, I'm interested to collaborate. And basically, that's why to attract students, I say I do tissue engineering. My questions are biological, but to make these things, we're actually doing tissue engineering because we're making the tissue to simulate natural tissue. So we have these tools in biomaterials and microfabrication. We build bioreactors to exercise or torture our cells, and we do a lot of imaging. And I'm looking for collaborators in these fields. So if you are interested, please let me know. Thank you.